Good afternoon, everyone. So my name is Fred Schumacher. I am currently the chair of the School of Medicine Lecture Committee. So we're people who take nominees. We then invite the nominees to come to give a talk and hopefully set up some meetings and have an opportunity to have some light fare and some drinks beforehand. So welcome today to the Lewis Bloomfield Memorial Medical Lecture. So this lecture series has been the oldest at Case Western that I could find. So I'm pretty new to this. I wasn't here when it started. I don't know if anyone was, but they were. It was 1955, all right? And this was a endowment that was started by the widow of Lewis A. Bloomfield, Kitty Bloomfield, and her son. And they donated what I could tell. This is an old document. It's very faded. I think it has coffee stains on it and everything. But they gave $10,000 to start this lectureship. And equivalently, that's about a $250,000 donation compared to today's dollars. And, you think about that, it was a pretty hefty donation at the time, but what it's really allowed us to do is have these opportunities to be able to invite some great speakers in. You can see it from the history of the ones we've had over the last 3 years, including the Lasker Award winner as well as a Nobel laureate, and then having Dr. Stu Orkin here today. This is a phenomenal chance for us to sit down from all different fields, from people from cancer biology to genetics to quantitative sciences and beyond, and medicine, have a chance to come together and hear this. And speaking briefly with Dr. Orkin earlier today, we feel like we're under assault a little bit sometimes from the outside pressures, so it's nice to be able to kind of come to an area where you feel pretty safe and have a chance to talk. So just to give a sense of the mandate of this endowment, was just an opportunity that the Bloomfields really wanted people to have a chance to bring in great lectures, to have talks with the medical community, the public, students, trainees, all the way through. So I think we've done a great job today. Thank you for being here. I'm going to have Dr. David Buckner come up and introduce Stu.
Perfect. Thanks, Fred. So it's a pleasure to welcome everyone here, both in person and those of you tuning in online, to the 2025 Lewis A. Bloomfield Memorial Lecture, presented this year by Dr. Stu Orkin. Dr. Orkin is the David G. Nathan Distinguished Professor of Pediatrics at Harvard Medical School and has been a Howard Hughes investigator since 1986. He earned his BS from MIT, his MD from Harvard. He trained for a few years at NIH under Phil Leader. Then completed a PEDS residency and PEDS Monk Fellowship at Boston Children's and Dana-Farber. His extraordinary contributions to science have been recognized with the Shaw Prize in the Life Sciences, the King Faisal Prize in Medicine, the Harrington Prize for Innovation in Medicine, placed on the Time 100 list of most influential people in 2024. He's an elected member of the National Academy of Sciences, National Academy of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society. He's perhaps best known for his pioneering work on hemoglobinopathies, which we'll speak about today. But I first encountered his work through his discovery of the von Willebrand's factor gene. So this was a science paper that he published in 1985. The first author of the paper, David Ginsburg, who can be seen on the right in the paper, and that's sort of Stu in the middle there from his lab in the 80s. Eventually became my postdoctoral advisor, making Dr. Orkin my scientific grandfather. So I still remember David's stories from the lab about cloning the BWF gene and how he credited Stu's creativity in pioneering the use of random hexamers to tackle that enormous gene. And so it's especially fitting to welcoming him for the Bloomfield Lecture, whose endowment was established as and this is a quote, as a permanent memorial in honor and recognition of Lewis A. Bloomfield as a father and a husband. And so for a lecture that's meant to honor family, it's a joy to host a member of my scientific family. So with all of these accolades, over 600 publications, a remarkable training tree that I think sets a record for the number of Howard Hughes investigators that came from his lab, it raises the question of sort of how do you really know that you've truly made it as a scientist? And I think when looking into Stu's work, I think I found the answer. And it's when JCI publishes an article that's titled "Stu Orkin is a Superhero." So this is a paper that was written by his former trainee Len Zahn. when he received the Cobra Medal from the Association of American Physicians. I highly recommend you check out the article. It's actually sort of half scientific tribute and half Comedy Central roast, I think. So it's a great article. But without further ado, it's a true pleasure to welcome this year's Bloomfield honoree, Dr. Stu Orkin, to share a bit about the science behind the superhero. Small token of our appreciation. So hopefully it fits in your luggage.
Thanks, David and Fred. I do feel like I'm coming back to a family here. So I am a grandfather to David. I don't know how he feels about that. Also, my daughter was here for 4 years with her husband. Her husband was a graduate of medical school, daughter of nursing school. And so I spent a fair bit of time in Cleveland. One thing you didn't mention in that list of honors was when I got listed as the Time 100, which was the craziest list I've ever been on. I was also there with Patrick Mahomes and Dua Lipa. Unfortunately, I couldn't go to the gathering. They had a Hollywood gathering in New York, but I had a conflict and couldn't go. My daughter offered to go in my place, but I didn't let her. So what I'd like to do today is tell you what we've done over the last more than 15 years and even longer, and try to give you a sense of where we are, but where I think we need to go, that the job isn't done, unfortunately, and tell you some of the interesting biology that we've encountered, which I think makes it even more interesting. So I think it's pretty obvious we're in a new era now in molecular medicine where new technologies are brought in for patients. And I think there's probably no better way to illustrate that than by the first approved editing therapy, which is Casgevy, which was approved almost 2 years ago now. And I'll tell you the backstory for that, and then tell you where I think we need to go, and also try to make some scientific points and perhaps some other broader points as we go along. So for my entire career, I've been interested in blood cells. I think I became interested as a medical student. I think largely because they were accessible. We could, take them out of the body and look at them on a slide. I wanted to know where they came from, how do they form, what happens in disease, and then lastly, can we do anything about it in terms of therapies? And much, but not all of our work has been in the area of red cells, and I think red cells, in a way, have been a fantastic experimental tool for this kind of endeavor, because there's a long history. Red cells really are a bag of hemoglobin, not much more than that, to carry hemoglobin around to bring oxygen throughout the body, deliver it. And hemoglobin was the first protein structure that was determined, and this is Max Perutz here with the Nobel Prize. Looking at this photo reminds me actually of the Three Stooges. I think it's just something about the way they're positioned on that model. But I particularly like this statement by late geneticist Arno Matulsky, who said that hemoglobin research plays a role in biochemical genetics similar to that of Drosophila research in formal genetics. And many fundamental concepts have been clarified by investigations on human hemoglobins. And this statement was made in 1974, that is before DNA cloning. And I think after cloning, the statement is even more prescient. And one way to illustrate that is from a review that Tom Maniotis had earlier, I think it was last year in Cell, or maybe it was this year, where he emphasized that the kinds of discoveries that really came from work that was really focused on either hemoglobin, red cells, or something related. cDNA cloning, one of the first cDNAs cloned was rabbit globin. Genomic cloning was really initiated largely in the field, and the recognition of split genes other than viruses really came from the study of globins. Comprehensive mutational analysis of disease we carried out with Haig Kazazian in the 1980s. First expression of eukaryotic genes in mice and cells, really globins as well. Super enhancers, which have been sort of renamed for those in oncology, is really discovered in the globin field, was the locus control region, and gene editing therapy has been applied in this field. So the field has been at the leading edge of molecular biology, and you can sort of trace the history of molecular medicine through these kinds of investigations. So besides the fundamental interest in sort of these genes and their expression, there are obviously two disorders which are prominent and worldwide affecting hemoglobin. One is sickle cell disease, was described more than 100 years ago, and this was by a cardiologist, actually, James Herrick. And then Linus Pauling, in 1949, termed this the first molecular disease. And then Vernon Ingram, 7 years later, identified the amino acid substitution in the beta chain of hemoglobin that is responsible for the disorder. And the other major disorder is beta thalassemia, which is not a disorder of the protein sequence, but one of production of beta globin. And these two are basically form the major hemoglobinopathies. Thalassemia on the left and sickle cell on the right. We've had the DNA sequence of the beta cluster, which is shown there on the short arm of chromosome 11. We've had the sequence of that since 1982. Staring at the sequence did not provide much insight into how the genes were really regulated. Both of these disorders are mutation of the adult chain, which is the beta chain. You can see a smear of thalassemia on the bottom left, and the various mutations, which are point mutants for the most part in the beta globin gene that affect production of beta globin. These are largely affecting housekeeping functions like RNA splicing and transcription, and coding potential, and sickle cell is a single mutation, one base pair out of 3 billion in the genome. So these are global disorders. There are now estimated 500,000 new births a year, largely of sickle. It used to be 300,000, but that number has gone up. In the United States, we have 75 to 100,000 individuals with sickle cell disease. The estimates are somewhere north of 5 million worldwide. I think a point that the late David Weatherall brought up was these are really global and disorders with a real health burden worldwide, which perhaps isn't greatly appreciated in this country. There are more in India, which I hadn't thought of much as a site of sickle cell disease. There are more than 1 million individuals at a minimum in India alone. And I think one aspect of distribution is they follow what's sort of referred to as the malaria belt in terms of incident. And Allison and Haldane, Allison was actually the major one who made this hypothesis, but then somehow it's also referred to the Haldane hypothesis that this tracked with malaria and that the heterozygotes have a selective advantage in the presence of malaria parasite. So this has really been the focus of the field for many years. think I'm getting a spam call. Yep, spam. I don't have a good spam blocker, I don't know what it is. So this has been the focus of the field and also the focus of our work for many years. And that is really, how are the genes expressed within this locus? There's an embryonic gene that's expressed in the first trimester of human development. There are 2 fetal genes, duplicated genes, that are expressed during most of fetal life. There are 2 adult genes, a minor 1 delta and the adult beta gene, which is mutated in sickle and thalassemia. And we really want to understand 2 things. Why are these expressed only in the red cell lineage? And second, why or how are they regulated in this developmental pattern? What causes this switching event? In all of us, we have about 1% fetal hemoglobin as adults, which is unevenly distributed in cells, so it isn't totally extinguished. Now, the problem in both sickle cell disease and in beta thalassemia is there's a hemoglobin problem. It's obvious. There have been Mendelian disorders with variable phenotypes depending on other genes that you might inherit. In terms of sickle cell disease, what kinds of options could we have to intervene? One would be to interfere with polymerization, and there have been many attempts to do that biochemically, even therapeutically. You can shift the oxygen dissociation curve, which was a drug from Voxator by Global Blood. It's been withdrawn for a variety of reasons, although it wasn't very effective to begin with. I don't know why it was ever approved. And you can change the hemoglobin. You can repair the sequence, you can provide a new hemoglobin, or you can switch back to hemoglobin F, which can replace adult hemoglobin. And the reason it can replace it is that the properties of hemoglobin F are different, but not so different from adult hemoglobin. So hemoglobin F is a tetramer of the alpha chains, which are in both hemoglobin F and hemoglobin A, the adult hemoglobin, but the difference is the gamma chain, the beta-like chain. And hemoglobin F has a slightly higher oxygen affinity, but it's not so much a problem that if any of us have 100% fetal hemoglobin, we're still perfectly fine. Hemoglobin F can replace hemoglobin A without adverse consequences. Even mothers who have 100% fetal hemoglobin can still undergo normal pregnancies and have normal offspring. So there's still good oxygenation of the fetus, even if the mother has hemoglobin F. Hemoglobin F is very good at blocking polymerization of sickle hemoglobin, particularly when you get higher concentrations. Hemoglobin F is subject to genetic control within families. That low level of 1% is genetically controlled. And then there are interesting genetic syndromes that are called persistence of fetal hemoglobin, or HPFH. There are common varieties that have small differences within families in the hemoglobin level. And then there are exceptional individuals, very rare, who have very large effects. They have 30% fetal hemoglobin from 1 point mutation. We'll come back to those. Those are beneficial or up mutants as opposed to down mutants. The story of fetal hemoglobin actually dates back to a report by Janet Watson, who was a pediatrician, didn't do any experiments at the bench. This paper was essentially just looking at some clinical data, phenotypes, and what she noticed was that newborns who were going to have sickle cell disease as they got older were not sick in utero or as in the neonatal period. And she surmised correctly that they had a high level of fetal hemoglobin, which hadn't yet been turned off. And so she proposed that hemoglobin F was protective. And in fact, she was right. This wasn't from any biochemical experiments. It's been supported by a tremendous amount of clinical data since. So the therapeutic vision that was brought on the scene really as soon as, really almost when I entered the field, was that if we could raise the fetal hemoglobin level from this 1% to maybe 20, 30%, you could call whatever threshold you might want, it would be enough to provide hemoglobin to those with thalassemia who had deficient hemoglobin, or enough in the case of sickle to replace some sickle hemoglobin and prevent the sickling process, which is the polymerization of hemoglobin. The question was how to do that. So this is really the crux of the problem, that the locus, the beta globin locus, looks like this, roughly the chromosome. This is the locus control region, which is the super enhancer. And it's required for the expression of all the downstream genes at any stage. And basically it loops to the downstream genes. There's a lot of evidence now from experiments that actually physically loops to the downstream genes. And in the fetal stage, the gamma genes are on and the beta gene is off, largely by default, because the LCR cannot engage that gene. And then something happens as you make the adult transition where the gamma genes are turned off and the beta gene then becomes available for the LCR. Again, largely by default. And the question was, how does this all work? This just gives you a view of this kind of looping and just summarizes the switch. The real question was, how many factors are involved? Do they act positively or negatively, and what can we learn about the mechanism? And this has been the crux of the problem in the field for a few decades. When early on, I think we assumed that there were many, many factors, that there would be a whole battery of factors that would be involved, so complex perhaps that we wouldn't be able to either sort it out, or each one would contribute only a little bit. And we thought that because we had trouble finding what factors might actually regulate this switch. And we'd spent actually in the 1990s to the early 2000s, a lot of us spent lots of energy trying to find factors that were stage specific, that might account for this switch, that would work directly somehow within the locus, and we came up dry. And at that point, we needed something new, and I love this quote from Marcel Proust: "The only real voyage of discovery consists not in seeking new landscapes, but in having new eyes." We needed new technology. And I think this is a recurring theme in sort of the march of science, that we need a new technique when we come to some barrier. And that new technique was genome-wide association studies, which are just coming on the scene. One study was performed in the UK by Swilethine. Another one was performed on the island of Sardinia, which is an island with lots of thalassemia at the time, and a rather homogeneous population, if you will. And the study was actually supported by the NIH. It was actually a study to look at cardiovascular markers, blood markers, et cetera. We eventually linked up with them. And in this case, it was to measure using SNPs across the entire genome. In this case, looking at hemoglobin F as a trait, and to determine what loci or what regions of the genome might correlate with the level of hemoglobin. This is what came out of that, the QTL by Swilethine, as well as this GWAS from Sardinia, hit on the gene B11A as a new player. There were really three loci that the GWAS hit. B11A on chromosome 2, the MIB gene on the long arm of chromosome 6, and the beta cluster itself, which we knew was a site of genetic variation. If you sum these, the whole genetic contribution was at least 50% to the level of, which is huge for these kinds of GWAS studies. And the contribution of B11A was really quite enormous. That's why I put the Empire State Building. And at the time, MIB, we suspected, was involved more in differentiation of cells, and that's turned out to be the case. But B11A was came out of left field, if you will. It was known at the time this was a gene that was involved in B cell development, if you knock it out in a mouse. And it had the name B11A because the B was for breakpoint, and it was from retroviral mutagenesis in mice. It actually could insert and cause a leukemia, and that's the way it got its name. Like BCL2, this is BCL11. Had nothing to do with cell death, but that's just historically the name. So we started focusing on BCL11A because it was a new candidate. There was strong genetics behind it. But the challenge is you start with a genome of 3 billion base pairs, and you want to get down to the needle in the haystack and try to figure out, well, what is that gene really doing? How is it relating? What is the genetic variation all about? I'll come back to this part of it after a little more about BCL11A. The initial evidence that it might have something to do with regulating fetal hemoglobin was a relatively simple experiment. First, we looked at the distribution of protein in this case in either primitive embryonic cells, fetal liver, or bone marrow. You can see the embryonic cells don't have the protein. Fetal liver has some, bone marrow had a lot more. The mouse is different because the mouse doesn't have a fetal stage, it only has embryonic and adult. So in the mouse, it's present only in the fetal and adult stages. So it's stage specific, which already gave us a clue it was likely to be correct and involved. We then used human blood progenitor cells, CD34, which you could differentiate in the laboratory into essentially red cells. And we did the simple experiment of using a hairpin, shRNA, to knock down BCL11A. And when you do that, you get more gamma chain as well as more fetal hemoglobin. We basically made the statement back in 2008 that this could be a therapeutic target for reactivation of hemoglobin F. That was a pretty bold statement based on this experiment, so, but in retrospect, it turned out to be correct. The real critical experiment in my mind, though, is this one, which is how important is this factor? In the field, people have identified other things they suspected to be regulators. In whatever field you work in, whatever you find, you want to make the top regulator or whatever you're looking at. The question is, how important is a single factor? And we then did this experiment, which was to use sickle cell mice. These are engineered mice made many years ago, which have sickle cell disease. These mice have no mouse globin genes. They have human globin genes in the place of the mouse genes. They're made by old-fashioned homologous recombination. They have sickle cell disease. They're anemic and they're not well. And we bred those to a conditional knockout for BCL11A, but in this case, inactivated BCL11A gene only in the erythroid lineage. We wanted to do that to avoid any confounding effects in other cell types in the body. We just wanted to see what the effect was in the erythroid lineage, and we did that with EPO receptor-driven Cre recombinase, standard sort of conventional mouse genetics. And really the question is, would we rescue these mice? And in fact, the result was much more striking than we even had imagined. Here's a sickle cell mice. Here's a normal mouse. The smear of the crossed mice is perfectly normal. The hematology is normal. The mice have lots of fetal hemoglobin, and it's in virtually all cells, which is the critical thing. The distribution is really quite homogeneous across cells. Another striking aspect of this that I think we appreciated, but it's unusual, is that this transcription factor in this lineage could be entirely knocked out, and yet the cells otherwise seemed relatively okay, which is about the only transcription factor we've worked on in which we could get away with this maneuver. And we have some thoughts about why this is the case. But anyhow, this experiment said that manipulation of this gene alone was sufficient to reverse the phenotype of sickle cell disease. And this experiment, which was done in 2011, really is the basis for most of the gene manipulation studies that are going on today and probably will go on for a number of years. There is one other factor that's involved. The other factor is LRF or ZBTB7A. This is a factor that I'll come to a little later. It's about as potent in terms of keeping F silent, so they're both repressors. It's not the physiologic regulator. It's not stage specific. So now we have really two factors that are involved in silencing the GABA genes. And we think these are the only factors. If you knock them out, as done in this unusual cell line, which is important in the field, if you knock them both out, you get 100% fetal hemoglobin. We don't think that there's any other direct regulator. There may be other things further upstream, which we'll come to. So how does B11A work if it's a physiologic regulator? Well, it's a big gene, a big protein, more than 100 kilodaltons. But at the C terminus, there are 3 zinc finger motifs, closely spaced. And these bind DNA, and they bind a specific sequence, TGA-CCA, which was unique in the transcription factor database. Here's the crystal structure of the fingers on DNA. Why was this such an interesting sequence? Well, it turns out if one looks at the gamma promoter, and they're duplicated, there are 2 genes, they're duplicated, they have the same sequence, so we're only showing 1. If you look upstream in the promoter, here's the sequence TGA-CCA, and there are naturally occurring hereditary persistence mutations. These are these up mutations. Most famous one is at this position, -117, a G to an A leads to about 30% fetal hemoglobin in an individual. How do we know that? We know that from 2 papers in 1985. You see, Francis Collins was the postdoc at the time of one of those papers. And these are so-called Greek hereditary persistence of fetal hemoglobin, exceedingly rare, but was recognized. They thought that the mutation affected the cat box, CCAAT, which is a ubiquitous transcription factor binding site, but it overlaps TGA-CCA. They had no reason to think TGA-CCA might have been important. This is the important sequence. So what these mutations, there are 2 other naturally occurring mutations. So what they do is they prevent B11A binding so the gene can't be repressed. That's simple. So how does it work? In the fetal stage, the LCR loops to the gamma gene. There's another CCAAT, a cat box, more proximal than this in the gamma promoter. That's bound by the transcription factor NFY and various co-activators. This drives transcription. The beta gene is off. And then what happens in the adult stage is B11A comes on. It's a stage-specific factor. It binds to TGA-CCA, evicts NFY from the promoter, recruits the NERD complex, shuts off gamma, and the beta gene is now on. So this is all local control over about 30 bases. So this developmental decision is really based on a very small genetic platform, despite a very large complex. So if you look, you blow up the gamma promoter a little more. Here's where NFY binds in the fetal stage. Here's where B11A binds in the adult stage. Out here, -200, there are HPS, these naturally occurring mutations that lead to persistence of fetal hemoglobin in the adult. There are naturally occurring point mutants in the -200 region, and that's where the second repressor, LRF, binds. They work more or less independently, although one could modify that statement. But they both interact with a repressive complex called NERD, a co-repressor complex. We'll come back to that. So the genetics tells us a lot, and there are a whole series of these point mutants that occur in the gamma promoter that are associated with increased fetal hemoglobin. They're the ones at the -200 that affect LRF. The ones here affect B11A. There's even a mutation in between that leads to new binding of a positive factor. And so basically it's really the competition between activators and repressors that ultimately determines the fate at this locus. And so now we can rationalize almost all the human mutations that have been reported over many years. Okay, want to return to the genetics. Said B11A came from genome-wide association, common variation. What is that all about? Well, if you stare at the B11A gene and do some chromatin kind of biology on it, here's the B11A gene. There's a very big intron within the gene, and there are 3 DNase hypersensitive sites in chromatin in erythroid cells, but not in other cell types that don't express B11A. And that cell-specific DNase hypersensitivity is a sign of a transcriptional enhancer. So this is an enhancer in erythroid cells. We took this enhancer, put it in transgenic mice, and it drives stage-specific, erythroid-specific expression in mice. And we recognize that this really was interesting for a number of reasons. One is that the erythroid expression of B11A was driven by a specific enhancer, which directs both stage and tissue specificity in transgenic mice. If one removes this enhancer, the expression of the gene decreases 100-fold. So it's absolutely dependent on this enhancer. And that told us that if we could do some sort of cell modification at this region, it would be genetically sort of a safe harbor, if you will, in cells. It would be restricted, the effects would be restricted to erythroid cells, even though the gene might be expressed and is expressed elsewhere. So B11A is expressed in other places. It's expressed in B cells where it's required, at least in mice in a knockout. It's required in hematopoietic stem cells in a way we don't really understand, but long-term sort of maintenance. It's expressed in breast tissue and is said to be important maybe in triple-negative breast cancer. I'm not sure that's entirely correct. It's definitely expressed in brain where it has a developmental role. But the question is, how specific is this enhancer? If you take the enhancer out in mice, the corresponding enhancer to the human, the mice are perfectly fine. They have no expression of B11A in erythroid cells, but they have expression in B cells in brain where the gene is also expressed. And they switch their hemoglobin pattern if you put in a human transgene as a marker. So you just flip, here's gamma shut off in the wild type embryonic mice, and here it's now basically persistent. So this enhancer is absolutely erythroid specific, and so we began to focus on that enhancer with the thought of doing gene editing at the time, which was really zinc finger gene editing and talon gene editing, which is sort of old forms of gene editing. And we proposed really that this rational intervention might mimic common genetic variation. In other words, by affecting this enhancer, we could boost fetal hemoglobin. Fortunately, about the time we made these observations, CRISPR came on the scene. And so we were rapid adopters and used CRISPR then to dissect the enhancer. And to do that, we did an unbiased screen of the entire enhancer region to try to see if there were particular regions within the enhancer that were particularly vulnerable. In other words, this enhancer, which is 13 kilobases in size, it would have been kind of a bold experiment to try to take out the entire enhancer for a therapeutic purpose. That didn't seem reasonable. We wanted to see if there was something that we could modify that might be more selective. And the experiment then was basically to do a pooled CRISPR screen. This is one of the first sort of unbiased CRISPR screens that was done for sort of gene regulation, if you will. We used cells that expressed Cas9, erythroid transformed cells selected for high F, and basically found a vulnerable site in this middle enhancer. And this is actually a blow-up of that we went from the original SNPs to some sentinel SNPs, identified the hypersensitive sites which are present in erythroid cells, but not brain or B cells, and targeted this region. And the guide RNA that's the most potent actually disrupts a GATA1 transcription factor binding site. That's the canonical master regulator for all erythroid expressed genes. We identified GATA1 in our lab in 1989, so really made a full circle back to this factor. This sequence here is actually the sequence that's used in Casgevy, the approved editing therapy. And it turned out that when CRISPR Therapeutics and then Vertex tried to improve on this sequence, they couldn't find a better guide. And this guide has no detectable off-target effects. This is what I call dumb luck. So this is actually the guide RNA that's used in the therapy. So we had gone from GWAS down to really understanding at a molecular level what the GWAS was all about, how the factor acts. BCL11A, I want to emphasize, is actually unique in that we can look at both common genetic variation and rare genetic variation, and they converge on this factor. I think that's why the genetics is so powerful in identifying this factor and really emphasizing its functional relevance. That you can modify the enhancer, it's modified through natural variation, and the binding site in the promoter is a site of genetic variation, but very rarely. So I think this convergence is really critical. So for those who don't know, how it's done, let me describe broadly how gene therapy is done, the hematopoietic system, and tell you specifically how it's done in this instance. So what's done is one takes from an individual, here an infant, although it wouldn't be the case in the first trials, hematopoietic stem and progenitor cells. These are essentially CD34 positive cells, only of which a small fraction are actually stem cells. Take these in the laboratory, they're modified either by lentiviruses in the case of viral gene therapy or editing, in case of editing therapy. You get a pool of cells that then has modified cells, hopefully a high fraction of modified cells. You have to condition the patient, usually with busulfan to eradicate the host bone marrow, then they're reinfused to the patient. There are many factors that are important to the execution of this kind of clinical maneuver. You want to know the fraction of cells that are modified, the efficiency of modification, the effect of the modification on hemoglobin F in this instance. What are the off-target or off-cell effects? What cell dose, preconditioning? All these are critical, and I just want to emphasize that this really builds on a tremendous amount of work from the bone marrow transplant as well as gene therapy field from years before. So in case of this particular approach, one takes from, let's say, an individual with sickle cell disease, the hematopoietic stem and progenitor cells, modify them at what I call the Achilles heel. This is a unique site that's recognized within the enhancer. These cells are reinfused into the patient. Hopefully the patient has high fetal hemoglobin and benefits. These are the first two patients who are treated by, in this case, by CRISPR Therapeutics at Vertex. Here's the guide again. This is an individual with beta transfusion-dependent beta thalassemia, essentially made no hemoglobin A. And this is a patient with sickle cell disease. Both of these patients, the first patients, were essentially functionally cured. The beta thalassemia patient, no longer transfusion dependent, basically has all hemoglobin F. and the sickle patient has on the order of about 40% fetal hemoglobin, sickle hemoglobin as well. And this was the subsequent trial which led to FDA approval. Here in the case of beta thalassemia, all these little dots are transfusions prior to therapy. And here after therapy, there are only a couple of patients that required some transfusions. Most have actually come off transfusions, more than 95% functionally cured. Same in sickle cell disease. These were hospitalizations for sickle cell crises, pain crises, and here afterwards were just a couple. So on the basis of this, which was really transformative in terms of clinical outcome, therapy was approved almost 2 years ago. And they've been rolling out slowly the therapy because it's ex vivo, it requires dedicated staff and obviously needs an experienced team, and they've established somewhere in the order of about 70 centers, both in the US and worldwide. There's another trial just to illustrate how general the sort of approach is. A colleague, David Williams at Boston Children's Hospital, used a different approach, which was to knock down B11A in an erythroid-expressed lentiviral construct. And basically, the therapy gives the same outcome, about 40% hemoglobin F. These are the number of F cells actually here, which is, you could calculate about 40% F. On these cells, and he's finished a phase 2 trial as well of that. This is actually the state of where the field is today. This has been approved, this is Casgevy. Bluebird Bio is a lentiviral therapy, which is just globin addition. It's providing a globin that doesn't sickle. It's been approved. It has the issues related to lentiviral delivery. The shRNA trial is ongoing. Editas Medicine had an interesting trial modifying the B11A binding site in the promoter, which is much more sensible than going to the enhancer. However, at the time when we initiated the studies, we didn't know the binding site, we knew thereafter. And that's what led Editas to modify the promoter binding site. That trial was very successful, was terminated because they couldn't find a partner to bring it to approval into the clinic. It's expensive to do the trials. Beam Therapeutics has a base editing trial where you don't cut the DNA, but you base edit the B11A binding site. That one's still ongoing and looks quite good. So I think the economics and the competitive landscape is going to affect when we see the next therapies approved. There are lots of advantages. I want to get on to some more recent stuff in the last few minutes, but there are a lot of advantages to increasing F, and I think one of the important ones is it's suitable for all the hemoglobinopathies, sickle cell disease, and the 50 different thalassemia mutations. They can all be dealt with by just raising fetal hemoglobin. How much do you need? That's always a question. This is a particularly interesting aspect of the genetics of this. I said that B11A is important in the brain in development. There's a rare syndrome of haploinsufficiency where one copy of B11A is either deleted or mutated, the other copy is normal. These children have an unfortunate syndrome of really a disability, really cognitive disability, autism-like syndrome, but the question we had was, what's the level of hemoglobin F in somebody with 50% the level of B11A? And the answer is really quite there. Okay, the level is about 13, 15%. It's a wide range, but that's 10 times normal with having 50%. So there's this would be beneficial in a patient with sickle cell disease. It's almost in the classical hereditary persistence of fetal hemoglobin range. So you don't have to turn the protein completely off. you can turn it down and get a big effect. And this is very important in thinking about, are there other ways that would be, that we could have a therapy that would be more widely distributed than an editing therapy. Just to emphasize how critical it is to the patients, this is actually the first patient who was treated, Victoria Bray, first one who had treatment by editing, and she's been very vocal and has been in a number of meetings, and here she basically says, you know, I had no life before and now I have a whole life. I mean, it's just remarkable, the transformation. So how can we get this therapy or these approaches to more individuals? One way is to reduce the preconditioning in the bone marrow, essentially the bone marrow transplant. There's a lot of work going on to try to do that, and the conditioning is toxic. In fact, a couple of patients, one in the BEAM trial and one, I think, Vertex more recently, have died from complications of preconditioning. So it is critical. One might be able to enrich cells in vivo after they've been transplanted back. There's a lot of work going on in that area. And then there's a question of doing in vivo gene therapy, where you give like a shot with a lipid nanoparticle and editing component. A lot of work going on in that area. I think the major challenge there is a quantitative one, that you have to modify at least half of the hematopoietic stem cells to get to therapeutic range. So is editing enough? No. No one thinks so. I think we need other approaches. Why is it not enough? The approach is labor intensive and complex. It takes about 6 to 9 months from entering the therapy until you're out of the hospital and well. It's expensive. It's $2.2 million, but I think that's a fantastic buy if it cures you and you don't have medical costs, and the family can now have normal, family environment, if you will. Patients can then work and have a livelihood. There are insufficient numbers of transplant centers. We have 75 to 100,000 patients in sickle cell patients in the U.S. 25,000 are dubbed severe, which would be eligible for therapy. There's no way we can do transplants of 25,000 individuals. Can't be done in parts of the world where most of the patients reside. And so we've actually advocated, as have some others, particularly Vertex, for a small molecule. Perhaps if we learn enough about the mechanism of switching, we can design small molecules which would be essentially targeted therapy, which could achieve the same result. And we know how to deliver pills worldwide. So our journey has been one of figuring out mechanisms of action. The other is genetic therapy. But now what we've been doing more recently is focusing on how could we ever conceive of using small molecules to target transcription. And I think many of you know that transcription factors are the worst targets. It goes to alpha fold. B7A is basically spaghetti. It's a very unstructured protein with a NERD binding domain at this end, DNA binding domain over there. And these proteins are bad because they don't have the right pockets, if you will, to take a small molecule for conventional pharmacology. One thing that a number of people have suggested, well, if this protein is so bad, why not go for a different protein, a different target that would be easier? This is the answer I give. There are only 2 targets that are worth anything. In my mind, these are the transcription factors that bind the DNA. They're the direct repressors. Neither one is easy to target with small molecules, and this is the physiologic regulator. All the other stuff that's actually drawn a lot of attention, and epigenetic regulators, which I haven't shown here, are far upstream. They have less bang for the buck, and they're more pleiotropic. They affect many other genes. And you have to get the repressors off the DNA if you're ever going to get the genes expressed. So my response to the question of why don't you go for something easier is there is nothing easier. And good luck if you want to follow the others. Okay, so I think one point I want to make in the last 2 minutes is B11A is thoroughly validated as a therapeutic target. There's almost no target that now is validated to this degree, genetically, biochemically in patients. And so we ought to focus on this. And last few minutes, I promise, just gonna, may run over 5 minutes, if that's all right. Tell you, this protein is really exceptional, but it's also telling us some really interesting things. I'll just give you the bottom line of some of the recent work. This protein has 7 zinc fingers. The first zinc finger, which is numbered 0 here for historical reasons, if you isolate it, it forms a tetramer, which I've never heard of before, a zinc finger forming a tetramer. Here's the structure of the, here's crystals, here's the structure of it, symmetric tetramer. Why is that important? And I won't show you the data that builds to it. You can go to a paper we had at the end of last year. As the protein is first expressed, zinc finger 0 is a monomer. The next finger, zinc finger 1, is a monomer. There's a degron in this finger, the degron sequence, which is recognized by proteins that want to destroy proteins in proteasome. In order for the protein to be stable, you have to form a tetramer and the dimer here to block the degron. Once you've done that, the protein is rock stable in the cell, half-life of the life of the cell. There's no monomer in the cell. It's an obligate tetramer. It's actually probably even a little more than a tetramer. So this is a big protein, and it has an unusual sort of biogenesis, if you will, in the cell. This is what the tetramer looks like if you print it out from a 3D printer. There are some little pockets there. So what's the endogenous, if there's a degron, what's the endogenous ligase that deals with that? I won't tell you how we got to it, but there's an F-box protein 38, which is the protein that acts on that. If you take that protein away, you can actually express this as a monomer here, and you get more protein. So it regulates the level of protein in the cell, steady state. So why does this multimer benefit the cell? Well, we've done experiments to get at that, comparing an engineered monomer, which you can do when you know enough about how the protein gets produced, versus a tetramer. And the monomer is not very good at repression. It's not very good at binding DNA, and basically it's not very good at cleaning out NFY from the promoter. So you basically need a big structure to maximally repress. So how do we think about that biochemically? There are now a number of ways to look at the kinetics of proteins within the nucleus. There are varieties. This one is used a lot as FRAP, where one uses a laser to bleach an area and then see how long does it take to come back. There are a number of other single molecule tracking systems. If you read the literature, what the literature says about transcription factors is they're highly dynamic in the nucleus. They're moving around all the time. They have very short residence time on chromatin on the order of seconds. And that's because transcription is stochastic, it's periodic. But these findings are all derived from a very small number of transcription factors, most of which are transcriptional activators. What we've done is look with these techniques at B11A, and this is the work of a postdoc, Nan Lu, who's now in China, using FRAP. Here's GATA1 and P53. You get photo bleaching, and then the signal comes back. B11A comes back very slowly, and this is H2B, which is essentially inactive, doesn't move. And he's looked at a whole bunch of transcription factors, both activators and repressors. And as a whole, the repressors are relatively slow mobility compared to the activators. And I think that makes a lot of sense. repressors really kind of slow down the system. You want to have long residence time on chromatin. So they are different. I just don't think it's been recognized before. And I'll skip this and just sort of say that what we believe happens now is that B11A is it's a complex, big protein, a tetramer. We think it's relatively stationary in the nucleus. It has a very long chromatin's residence time by these kinds of assays. And it recruits a lot of co-repressor. And what it does is it counteracts the effects of the locus control region, which is a super enhancer that's meant to sort of drive the globin gene. We also know that this structure is affected by phosphorylation. If one dephosphorylates this, the protein becomes more mobile, falls off the chromatin, and no longer can repress. So we now have a lot of thoughts about ways to interfere with B11A with small molecules. One would be to affect the way in which the tetramer is formed, or the stability of the tetramer, use the tetramer for protein degradation, affect the degron, this dimer that occurs. And lastly, we could use protein phosphorylation to change the status of the protein. So there are a number of avenues to pursue, so I don't think it's an entirely hopeless endeavor to try to get small molecules. So in conclusion, just 2 slides include, it's an obligate multimer in cells. There are unstructured regions that would contribute to this highly ordered structure. Native B11A is atypical for a transcription factor. It's really not mobile at all. And this property of immobility is critical to repressor function. And we think it counteracts an enhancer function, and that's why it really has to be there all the time. And mobile state is observed for an engineered monomer, and mobilization is another way to be a vulnerability for repression. So we have a number of ways to look at interfering here with the tetramer, targeting things to the tetramer, interfering with the dimer here, enhancing this interaction with a ligase, perhaps with a glue, or mobilizing the multimers. And we're exploring those. We don't know if any will work, but that's the current work we're doing. So in conclusion, through human genetics, molecular biology, I think the basis of the transcriptional switch from fetal to adult globin is now understood in really molecular terms. And I think we can confidently state that B11A is the physiologic regulator. There is a second repressor, but it's not the physiologic one. And taking advantage of that, we know how to reverse the switch now, which has led to the editing therapy, and I think will lead to 2nd and 3rd generation ones, and possibly contribute to in vivo therapies later. Only a small number of patients will receive the current therapies because of the cumbersome nature and the infrastructure needed. So I think the imperative now is to use this understanding for novel therapies. Spam again. Yeah, better spam blocker. So a few just take-home messages maybe for the trainees. Human genetics provides invaluable cures and clues to things. I don't think this problem could have been worked out without the human genetic contributions. And I think it's really important to think of therapeutic targets in these terms. The development of novel approaches requires a convergence of several paths, often far afield. I mean, GWAS came on the scene at the right time, CRISPR came on the scene at the right time. Identifying targets is easy, translating them is hard. It requires a lot of luck and things coming together. I would argue that deep mechanistic interrogation can provide new insights into unforeseen pathways, both in biology and in therapeutic intervention. And I guess, if nothing else, persistence. A number of people contributed to the early studies shown here, particularly Vijay Sangran, who's now a professor in our division of children's, and Dan Bauer, who's a leader in editing therapy for hematopoietic disorders. And the more recent contributors are shown here, those who contributed to the protein assembly story, ligase, and nuclear dynamics. And I'll take any questions.
Fantastic. Wonderful career. I have a question about, are you familiar with the molecular degrader for WIZ that was discovered by Novartis? And
Yeah.
I imagine they were probably looking for BCL11. When they did that screening, they didn't find it. What are your thoughts on that drug and that as
Yeah, this there are actually two companies that have, the first company was Novartis, reported a molecular glue, an imide-like molecule that gets a protein called WIZ. I didn't mention, the B11A fingers look like Icarus fingers, so you'd think they would bind imids. To my knowledge, no one's found imids that react with the former complex. So Novartis found WIZ. BMS has another glue which gets both LRF and WIZ. And they're both said to be going forward clinic. My issue with WIZ is there's absolutely no mechanistic basis for WIZ being involved in this whole process. It actually is part of G9A complex kind of function, and that has been explored with small molecules and actually by Epizyme and failed. But I don't think they're gonna get enough bang for the buck, because I think it's very far upstream. And WIZ also interacts with CTCF sites, and I think something like that smells like it's gonna get them into side effects.
Yeah.
And one of the things you have to recognize for these disorders is if you give chronic therapy, it needs to be a pretty safe drug. I don't think that'll do it. The WIZ LRF degrader might be a little better, but we don't know what the side effects are gonna be, especially if you combine those two.
Sure.
It may be better, it might be worse. So I don't think we know the answer, we'll find out. They were, I'm sure, looking for, as everybody has been. Yeah.
Great talk, Dr. Orkin. I had a question on your therapeutic approach for genomic editing. Why are you trying to promote fetal hemoglobin versus just correcting the mutation?
Yeah, that's a very good question. I think why go for that? Part of it is historical, that the first editing approaches were very, very inefficient and bad at correcting the sickle mutation. So the approach now that you would propose to use is like prime editing from David Liu, who spoke last year. Even prime editing, the efficiency, it's not that fabulous. And I would argue, if you can raise F, you can take care of all thalassemias too, so why not have a one-stop shop? So I'm not sure that there's an enormous advantage to correcting the sickle mutation other than, yeah, it would be more elegant to do so. Sort of like the question of why we didn't knock out the B11A binding site rather than, instead of the enhancer. At the time, the enhancer region was identified and everything was kind of on the pipeline. We didn't know the binding site. We only found it out shortly thereafter. So part of it's historical.
Thanks for the talk. I just have a question. So does that mean that when you increase the percentage of F, does that mean when you increase the percentage of F, it's going to suppress S, or S will also keep coming up?
One thing I didn't, sorry, it's a good point. One thing I didn't emphasize is that if you increase F or a gamma expression, the sickle expression goes down because the LCR can only contact one gene at a time. So it's reciprocal. So everything you do to increase F reduces sickle, plus the F protein inhibits sickling process. So you get two benefits.
All right, I think we'll take one last question then from Lamar.
Can you block the binding site, like using an antisense oligo type of approach?
Yeah, you might be able to block the binding. Remember, the affinity of binding is on nanomolar, so it'd be very hard to do. And also, you need chronic administration. I don't think that's practical. But in principle, you might.
Much easier though.
Your
So,
question, Stan?
Stu, I think also longer term survival of the F red cell.
Yes.
So,
Yeah.
another thing that's really big. Given the locus, why hasn't there been, or has it failed to think about peptides that block other protein interactions with BCL?
Peptides, there hasn't been a lot of work on that. There has not been. I mean, from a therapeutic standpoint, you'd have to give them all the time.
Yeah, I got it.
So, I don't know, we have nanobodies, for example, which bind B11A, and we can use them for protein degradation, but I don't know how you would make that into a therapy. So, I guess my thought is focus on the things that we might be able to translate.
All right, well, just a reminder that the reception will continue after the talk is over.